have your blood analysis, you can turn it in now because it's due tomorrow. And I'm missing some endocrine things from people. Okay. Megan, you need to bring yours, when you get back. You have to do the endocrine thing and the blood thing. Okay? Even though it's only 5 points, you have to do it. So do it. Okay. All right. About so far. We've talked about erythrocytes. You have about 5 million erythrocytes. They are biconcave discs with a little area of central pallor in the middle. It takes them about a week to develop, and then they will last for about 120 days, which is about 4 months. What they do is they transport oxygen and carbon dioxide, by far the most important one being oxygen. And they do that by being little bags of hemoglobin. Other white blood or other formed elements in the blood. You have your white blood cells, also called leukocytes. Typically, you have about 10,000 leukocytes per microliter in the blood. Backing up a second, just to put some more numbers here. Okay. You have about 5 million per microliter of red blood cells. You have your normal hemoglobin is 12 to 16 milligrams per deciliter, and then you have your hematocrit, which is approximately 45%. And all of those are numbers that I want you to know. Okay. For the white blood cells, I want you to know that you have about 10,000 or 10K. Okay. Now, if you would like to memorize the numbers of these other of each type of white blood cell, you may, but I basically want you to know relatively how many of them you have. So, for example, by far the most white blood cells that you have are usually neutrophils. Neutrophils are granulocytes. They are also called polymorphonuclear cells, or polys. That's because of the way the nucleus looks. It looks like it's almost pinched off and divided into a bunch of little pieces. Neutrophils are specialized for phagocytizing bacteria. The other granulocytes are eosinophils and basophils, and you have far, far less of them. Far, far less of them. Eosinophils kind of stain reddish because they stain with eosin. The nucleus kind of looks like it's bilobed or divided into 2 pieces. The granules are usually very thick. And it's specialized for killing parasites and for inactivating things from the immune system, or byproducts from the immune system. So you see a lot of eosinophils with parasitic infection and with allergic reactions. Basophils usually stain very, very darkly. They almost look black sometimes. They also have a bilobed nucleus, but sometimes you can't even see it because the granules stain so dark. There are even fewer basophils than eosinophils. They release histamine and they release heparin, and they are also involved in the inflammatory reaction. Now, when we go over the inflammatory reaction as part of the immune system here in a few weeks, there's a bazillion different chemicals that are involved with the immune system. One of the places that those chemicals come from, among many, is your basophils. Okay, the agranulocytes. You have your lymphocytes. Lymphocytes tend to look smaller, and they look like they have this nice round big nucleus with very little cytoplasm around the sides. Lymphocyte numbers are usually second only to neutrophils. In other words, you have the most neutrophils and then the next most lymphocytes. Lymphocytes are specialized for dealing with abnormal cells and viruses. They're specialized for dealing with abnormal cells and viruses. Lymphocytes are also the cells that produce antibodies. So abnormal cells, viruses, antibodies. We're going to go into a lot more detail with lymphocytes and the different types of lymphocytes when we get to the immune system. The next type of agranulocyte is the monocyte. Monocytes are usually pretty easy to pick up because they have this huge curved nucleus, and the whole cell is bigger than these other cells. They're bigger. These cells are called monocytes when they are circulating in the blood. And there's not that many of them circulating in the blood because they go to where the problem is. The problem's not usually in the blood. So they will go to where there's a problem, they will crawl out of the blood, and then they will start moving through the tissue spaces. When they are out of the blood, monocytes are called macrophages. And with a name like macrophages, that tells you exactly what it does. Okay? They do massive amounts of phagocytosis. They are the garbage trucks, the Pac-Men of the body. They go out and they eat up everything. Okay? They can eat up debris, they can eat up bacteria, they can eat up viruses that have been coated with antibodies. Okay? They can eat up antibody-antigen complexes. Okay? They do a lot. The last cell type here, which we haven't talked about much yet, is the platelets or the thrombocytes. Platelets or the thrombocytes. You usually have about 500,000 to 250,000 platelets. These last a fairly short amount of time, and they help out with blood clotting. Now, when you are looking at a blood smear, what platelets look like are little tiny purple pieces of junk on the slide. They almost look like debris on the slide. Alright, so that's more or less, I think, where we left off last time, right?
Dr. Scott, what is this, and I know you said it before, I'm sorry, but what is the measurement that they just did? It's cells, and then
On that particular thing,
Yeah.
it was per microliter.
micrometer, and that's what the weird little U thing is?
Yeah. Alright. Platelets used to be called thrombocytes. Thrombo refers to clotting, okay? And they are involved in clotting. So calling them thrombocytes does make sense to a degree. However, platelets are not cytes. They are not cells. They are cell fragments. Remember that all these different formed elements develop in the bone marrow from your hemocytoblasts or your stem cells. And they develop into whatever type of blood cell is needed. If you need red blood cells, you make red blood cells. If you need platelets, you make megakaryocytes. If you need white blood cells, you make leukocytes. With a name like megakaryocyte, karyo refers to chromosomes. So megakaryocytes are not only huge, they have more than one set of chromosomes in their nucleus, which is unusual. So megakaryocytes are huge. They have more than one set of chromosomes in their nucleus. And they sort of shatter into pieces, which are called platelets. Now, I like this picture because it's easy. Some of the other pictures in your book, okay, you look at it and your brain goes, ah! Because there's a whole bunch of stuff on the picture. So let's look at the forest, and then we'll look at the trees a little bit here, okay? Now, when you damage tissue, the little cells in the tissue will scream. Anybody know the book 40 Years of Who? Instead of having little out there, what your tissues do to scream bloody murder is release chemicals. Damaged tissues release chemicals. The first thing that those chemicals do is cause all the blood vessels in that area to clamp down or spasm. So initially, you might have quite a bit of damage, but you may not have a whole lot of bleeding right away, because blood vessels try to keep your blood from leaking out all over the floor by spasming. Now, your platelets flow along through the blood, and they don't cause any problem. They just go with the flow, unless they encounter a rough patch in the wall of the vessel, or unless they encounter a chemical signal. Platelets bounce along, just chilling out, unless they encounter a rough spot in the wall, a tear, or some other abnormality in the wall of the blood vessel, or unless they encounter a chemical. If they encounter a torn wall or a torn blood vessel, or they encounter a chemical, they change and become sticky. Instead of going with the flow, now they stick. They stick to each other, they stick to the wall, they stick to everything. Your blood vessel spasm is being caused by smooth muscle clamping those vessels closed. Smooth muscle can't continue to do that forever, but hopefully your platelets have come along and they have gotten sticky, and now they're sticking all over the place. It's like putting a chewing gum patch on something. Now, the platelets activate what's called the clotting cascade, or the clotting mechanism. Now, the clotting proteins are a whole group of proteins that circulate in the blood. They're made by the liver. The clotting mechanism also includes things like vitamin K, and calcium. And when they were first discovering proteins in the blood, they would find a protein and they didn't know what it did, so they would just give it a number. They would give it a Roman numeral number. And then they'd find another protein, give it the next Roman numeral number. So they ended up with this whole pile of proteins, and they all had Roman numeral numbers, but they didn't know what they did yet. When they figured out what they did, each protein was interacting with another protein, but they had them named out of order. And this has made generations of students crazy. Now, the way that the clotting mechanism works is via a cascade. Now, back in the days before there was a whole lot of DVDs and a whole lot of internet, and before your mom let you watch very much television, and it was really rainy and nasty outside, you had to come up with things to do. And one of the things that we used to do is build domino things all over the room. Okay. You stand dominoes end on end, okay, and then you flip the first domino, and it all goes all over the room. Okay. Well, imagine if you have one domino here, and you have it so that that one domino will hit two dominoes. And then those two dominoes hit two more, which hit two more, which hit two more. Now you've got exponential dominoes falling all over the room. Okay. That's what the clotting mechanism does. Once you get it started, it's a cascade. Each flips the next one, which flips the next one, which flips the next one, and pretty soon you've got this whole big lot of chemical reactions going on. Okay. At the end of the process, the business end of the process, you have a protein called prothrombin. Prothrombin is converted to thrombin. Thrombin converts fibrinogen to fibrin. Now, with a name like fibrin, what do you suppose the shape of that protein is? Like fibers. Fibrin forms like a net, like a fish net. So when you have fibrin, all of the formed elements that are coming past are now going to get stuck in that net. If they're stuck in the net, can they leak out all over the floor? No, okay? So this is called blood clot formation, where you have all of the red blood cells caught in a net. Okay. So they turn into a semi-solid, and they're not leaking out all over the floor. Red blood cells don't actually change, but you no longer just have a liquid. Okay. All right? That's the big picture. Everybody with me? All right, for those of you who are nurses, measuring prothrombin to thrombin, this is called the PT, which you see on your clotting studies. Okay. Your PTT is measuring this part. Okay. Your INR is measuring this part. So everybody with me on the big picture. Okay, now let's look at the itty details. See what I mean? Now, I would never make you memorize every step in this because I've never been able to make myself memorize every step in it. It's like, I don't care. Okay, if you're a hematologist, you care and you memorize all this stuff. Okay, the rest of us, mere mortals, can just go look it up if we need to. Okay. There are several things, though, about this that you need to know. Okay. Remember I said that you could get clotting started more than one way? Okay. You could have the endothelium, which is the lining of your blood vessels, rupture. So there's a hole or there's a raggedy spot. That can activate your platelets. If you do it that way, that's called the intrinsic pathway for clotting. Now, you may have wondered, how come people who have heart disease and so forth, how come they get clots? This is why they get clots, because they've got junk in the endothelium of their blood vessels. Cholesterol, inflammatory stuff in the walls, plaques in the walls of their blood vessels. So when the platelets come along, the platelets go, maybe we should stick to that, okay? And that causes the clotting cascade to start, and you get a clot there in that little blood vessel. Okay. Can you have intrinsic pathway that happens when you are injured? Of course, if you tear blood vessels. With the extrinsic pathway, the tissue cells release a chemical called tissue factor, and tissue factor activates the platelets. Tissue factor can also activate the rest of these clotting proteins on their own. Now, when you have placental damage, sometimes you have tissue factor that's secreted from the placenta into the bloodstream. What's that going to cause the mother's blood to do? Clot. Okay? And you will usually just be a little bit of clot here, and a little bit of clot there, and a little bit of clot over there, until all the clotting proteins are used up. That's called DIC, or disseminated intravascular coagulation. In other words, it's disseminated all over the place. Mm-hmm. It's happening in the blood vessels, it's intravascular, and it's coagulation. It's also called a consumptive coagulopathy. In other words, it's pathogenic, and it's consuming all of your clotting proteins. Now, if you understand that, you can understand why people with DIC tend to bleed to death. People with DIC tend to bleed to death. At first, I was thinking, how come they're bleeding to death if they've got clots all over the place? Well, they've got clots all over the place because they've used up all their clotting proteins, and now they can't stop the bleeding. DIC is sometimes a problem in pregnancy, especially associated with eclampsia. Okay. So it's something that you are going to be hearing about. It's the intrinsic pathway there that you're involved in. You're releasing a chemical that causes it. It's not necessarily trauma, okay, that's causing it. Okay. Now, your dominoes here, okay, you've got 12 to 12A, which converts 11 to 11A, which converts to 9 to 9A, and then 9A converts 8 to 8A, which converts 9 to 8A complex. Okay, is this giving you a headache yet? Yeah. Okay, it's giving me a headache. All right, notice that calcium is involved here. So calcium is involved in producing clots. Remember we said calcium, you're going to hear about that until you're sick of it. Okay, one of the reasons that your body has to maintain a normal blood calcium level is so that your muscles work right. Another reason is so that your blood clots properly. Okay? Another reason is because calcium is a second messenger for a lot of endocrine hormones. Okay, now what do you suppose happens if one of these proteins here is missing?
Your blood doesn't clot.
Your blood doesn't clot. Have you ever heard of hemophilia? Okay, with hemophilia, it's factor 8 that's missing. There are other types of hemophilias. Okay, von Willebrand's disease is one. Okay, that involve other clotting proteins here. The most common one is hemophilia A, which involves factor 8. Now, most of these are genetic diseases where the person lacks the correct DNA sequence to make factor VIII. All right, eventually you get down here and your two pathways join, and the end of your process is prothrombin is converted to thrombin. Thrombin causes fibrinogen to be converted to fibrin, and then you have your net in place. You have your plot. Everybody with me?
Yep.
Piece of cake.
What was the I in the
Intravascular. Okay. So here you have an injury to your vessel that exposes collagen fibers. In other words, your vessel wall is torn, it's no longer smooth. Okay. First thing that happens is your vessel clamps down, spasms. Second thing that happens is your platelets stick to that. Okay. The platelets activate other platelets and they activate clotting proteins in the area. Your tissues around here are going to also release tissue factor. Both of those things are going to cause coagulation to occur. The business end of coagulation here is prothrombin to thrombin, fibrinogen to fibrin. The fibrin forms a net across your hole, okay? And so the blood vessels and stuff can't leak out. Now, can fluid still leak out here? Yes, okay, you can have fluid that leaks out there until you get the whole mesh clogged with red blood cells, and then you don't have so much fluid leaking out. Once you have the whole mesh clogged with red blood cells, the mesh can tighten down and sort of pull the edges of the wound together. Okay. Eventually, fibroblasts will migrate into that area and start laying down collagen, and then you'll have a repair going on. At that point, do you need your net? No. So you want to dissolve your net, correct? When you dissolve your net, you dissolve the fibrin. Picture this? No, not picture this. Okay. The chemical that dissolves the fibrin is plasminogen. Plasminogen is converted to plasmin, and plasmin dissolves your fibrin. Okay. Plasminogen is converted to plasmin, and plasmin dissolves your fibrin. Okay, that's how you get rid of it.
Where is the plasminogen?
It comes from the tissues. Okay. Well, actually, it comes from the blood, and then you have tissue plasminogen activator. But anyway, it's there. Okay. All right. So, if you want to activate this process, you use something called TPA, or tissue plasminogen activator. And there are bacteria that produce this. Your body produces it, but there are also bacteria that produces it. So, there's a whole bunch of them. Now, supposing that you're not bleeding to death on the floor, okay, but your blood is still clotting, and it's clotting in your cardiovasculature, your cardiac circulation. If you have clots in your cardiac circulation, it's eventually going to kill your heart muscle, right? Supposing you have clots in your brain. Eventually that's going to kill brain tissue. So it would be nice to get rid of that clot, correct? Now, when you have a stuffed up sink that's not draining, there's a couple of ways that you can deal with that, correct? What do? Okay, you can stick a snake in there and shove the stuff until it goes on through. Okay? Break it up until it washes on through. What else can you do? Okay, you can stick Drano in it, which is going to dissolve it. Okay. You can do both with clots, but the best thing to do with clots is to use Drano. Okay. TPA is your Drano. Now, have you ever heard that with strokes and with heart attacks, you have a short amount of time to get treatment before you have permanent damage? This is why. You have to get the Drano in there and get that clot broken up before the tissues downstream die. Okay? So, should somebody sit at home thinking that they're having a heart attack and go, oh, you know, we don't want to go to the doctor, it's so far to the ER, maybe I just have indigestion, you know, I'll just sit here and wait and see what happens. Not a good idea. Why? Because if it is a heart attack, you're having progressive tissue damage that then can't be reversed. Okay? Same with a stroke. Now, there's one other kind of stroke, though, where the blood vessels in the brain burst. If you have blood vessels in the brain burst, you're going to have bleeding out into the brain tissue, which is going to damage the brain tissue. So one thing that can damage the brain tissue, one thing that can cause a stroke is a clot in a blood vessel. Another thing that can damage brain tissue and cause a stroke is bleeding. Now, if a person is having a stroke because they're bleeding inside their head, do you want to give them Drano? No, why not? It's going to make it worse and you're probably going to kill them. Okay, so one of the things that they have to have the time for is to do the neurological studies, okay, the MRIs and so forth, to figure out exactly what is going on inside that person's head so they can give them the right stuff so they don't kill them. Okay. Does that make sense? All right, move the lights, Corey. All right. Sonny, I got that email and I haven't had a chance to read it yet.
Oh, okay.
All right, now, blood type. Okay. Is blood type of interest to midwives?
Yes.
Yes. A couple different reasons. Now, if you remember all the way back to chapter 3, where we talked about cells, and we talked about the plasma membrane on the outside of cells, remember we said that sticking through that plasma membrane, you had proteins and you had glycoproteins, or proteins with carbohydrate hanging on them. And remember that some of those proteins functioned in cell-to-cell recognition. Now, some of you probably live in housing suburbs or housing developments where all the houses pretty much look alike. Okay. What do you do to give people instructions to your house? What do you do to make that house look like it belongs to you?
They're numbered or decorated.
Okay, they're numbered, they're decorated. Okay, one of the ways that people decorate their houses sometimes is by sticking a flag out front. Either an American flag, or a flag with a kitten on it, or a pumpkin, or whatever. Okay, everybody got the picture? Okay, your cells do the same thing. your cells stick glycoproteins out there to identify them. You know that if the cells don't belong to you, your immune system will take the opportunity to take them out. Go to sleep, kid.
Yeah.
Your immune system will take out abnormal cells and foreign cells. How does your immune system know that they're abnormal or that they're foreign? They don't have the right flags on them. Okay. Now, there are lots and lots of different types of flags on the surface of cells. Okay. When you do blood typing, okay, there's like 15 or 16 different markers that they look at on the surface of the cell to get a as close as possible type match. Okay. The match is almost never perfect. So most people that are receiving blood, unless they're receiving their own blood, tend to have a reaction. Okay, a transfusion reaction, and usually it's a fever. Okay. But you're trying to minimize that transfusion reaction because if you had to give this person blood, they're hurting anyway, right? You don't need your immune system tearing things up on top of that. So you try to get as close a match as possible with those flags on the surface. Now there are two types of flags that seem to be the most important with transfusion reactions. There are two families of flags that tend to be the most important when you're talking about transfusion reactions. The first flag family is the ABO family. The second family of flags is the RH family. RH comes from rhesus monkey because they actually found this flag by looking at monkey blood. Turns out people have it too. ABO, I don't know where that came from. You have a bazillion other flag families like Kel and what's the D one? Delta and Kel, and there's a whole bunch of them. So, the first one that we're going to talk about is the ABO system. So, here's your blood cells. And thinking about the types of flags that you could have on your front porch here, you could have an A flag, right? You could have a B flag. You could have both an A flag and a B flag. Or maybe you don't like the AB flags at all and you don't have any on your porch. Okay. So what blood type would you call this one?
A.
Type A. What blood type would you call that one?
Type B.
Type B. What blood type would you call that one?
AB.
Type AB. And what blood type would you call that one? Type O. Now, for reasons that we're not going to get into here, your body develops antibodies against any of these flags that it doesn't have. Your body develops antibodies against any of these flags that it does not have. And with the ABO system, it will do that whether you've ever been exposed to those foreign flags or not.
How does it know?
That's still under discussion. Okay, and the information that's coming out, it appears that the gut's involved in that. Okay. So, with type A, your antibodies would be against what? You would have anti-B antibodies, correct?
Mm-hmm.
With this one, what kind of antibodies would you have? You would have anti-A antibodies. Here, you wouldn't have, because you've got both kinds. You wouldn't have antibodies attacking those. Here, what would you have?
AB.
You would have anti-A and anti-B antibodies. Everybody with me? Is this hard yet? Say no. No. All right. Now, all you have to do is a little bit of thinking here then to know what kind of blood you can give each person. Okay. With this person, what kind of blood can you give them?
A and O.
Okay. You can give them type A. You don't want to give them something that they're going to attack, that they have the antibodies for attacking. So are you going to give them type B blood?
No.
Are you going to give them type AB blood?
Mm-hmm.
No. So you're going to give them type A and you're going to give them type O. Okay. Here, what type blood are you going to give them?
B and O.
Type B and type O. Okay. Here, what can you give them?
Yeah.
You can give them type AB.
Type A.
You can give them type A.
Type B and type O.
You can give them type B. And you can give them type O. Because they don't have any antibodies here that they're going to be attacking. Or that they're going to use to attack. Wait a minute. Right, what are you going to give this guy?
Only type O.
You're going to give this guy type O. Now, if you notice here, one of these types of people can receive any kind of blood. The other type of person can give anybody else blood. This guy here can receive blood from anybody, right? So this guy is called the universal recipient. That would make this guy the universal donor.
How do the antibodies not go with?
The antibodies do go with.
So then how do they not?
Okay. So what she's asking is, if I give this person here type O blood, the person's immune system is not going to be attacking that type O blood. Correct? But that type O blood is going to have anti-A antibodies in it. Is that type O blood going to attack the person's blood? Yes, to a small degree. Okay. Hopefully, the amount of blood that you're giving is a small amount relative to the amount of blood volume that they have. Okay. Now, remember, I just said a few minutes ago that people almost always have a transfusion reaction. That's part of the reason why. Okay. You almost never have a complete match. And the further apart the match is, the more likely you are to have a transfusion reaction, either because of your body attacking or that blood attacking you. Okay.
Unless you're O negative, because you have to have an exact perfect match.
Okay. All right, so everybody understand that? Now, what happens, by the way, my kid, when he was about 5, I took him to the beach, and I took him up here to Jacksonville to, we were at Big Talbot Island up there, towards the river where there's a bunch of oyster beds. And I said, yeah, he's running around barefoot. And I said, Luke, you don't want to run on the oyster beds, you'll cut your feet. Okay, so what does he do? Runs and cuts his foot. Okay. So I take him over to where there's clear water, stick his foot in, wash the blood off his foot. And this kid absolutely went bananas. He went crazy. Okay. Like, just absolutely panicked. Like, what's the matter with you? And then I remembered my developmental psychology. Okay. The reason why you put Band-Aids on kids half the time is not because they really need a Band-Aid. It's because they're not sure where their boundaries are in relation to everybody else. Okay. And it makes them feel better. When they see body parts going away, it scares them. Okay. And that's what was happening with Luke. Right. I said, Luke, why are you flipping out here? Well, Mommy, my blood, I'm bleeding. And I'm like, do you know how much blood you have, Luke? No, but it's all bleeding out. No. Okay. You're leaking a little bit. Okay. So how much blood volume do you have? And this is another number I want you to know. You have about 5 liters. Now, somebody that's tiny is going to have some more or less blood. Somebody who's big is going to have more. But you have about 5 liters. A unit of blood is about 500 cc's. So, the more blood you have lost, the more blood you're going to be having to replace, and the more antibodies you would be replacing with it. Correct?
Mm-hmm.
So, the more blood you lose, the closer your match needs to be to avoid a reaction. Yeah. Okay. Now, there's some things that they can do about this. Okay? They can give antihistamines and steroids and things like that to minimize it. If you're going to have somebody die on you, you give the blood and then worry about it later, usually. Okay. And usually, when in a trauma situation, you're more worried about keeping their blood pressure up than you are whether their hematocrit's a little low. Okay. Now, I have seen situations where, trauma situations where they gave people 26 units of blood. Okay. What was happening in that case? They were bleeding out faster than you could get it in. Okay. In those sorts of situations, it doesn't even. You may as well give them type O blood. You're just trying to keep them alive, okay, till you can get them sewed back together. Okay. All right. So everybody understand this?
Mm-hmm.
Okay. Is this something that you could turn your paper over and write on the back of your paper?
Mm-hmm.
Yeah. Okay. So you should be able to figure out any blood question that you are asked by doing this. Now, do antibodies cross the placenta? Yes, they do. Yes. Okay. What happens if you have a mismatch between mom's blood type and the baby's blood type? The mom already has antibodies if we're talking about the ABO system. Okay. So why don't the mother's antibodies attack the baby's blood? The short answer to that is sometimes they do. They can. It's usually not a big problem with the ABO system, usually. With the RH system, it's a big problem. So we'll talk about the RH system here next. It's usually not as big a deal with the ABO system. With the RH system, it is a big deal. Okay? But you can have the mother's blood attacking the baby's blood in both cases. Okay? All right, can I erase this? All right. So let's talk about RH because it's a little bit different. With the RH system, you have a flag that says RH. So what type blood would this be?
Positive.
RH positive. This one has no RH flag. So what type would it be?
Negative.
RH negative. So far, it's the same thing that we talked about before. The difference comes in the antibodies. Here, will there be any anti-RH antibodies? No. Why?
Because you need exposure to develop the antibodies.
No. Why does this one have no anti-RH antibodies?
Because it has that protein.
Because it doesn't, you don't develop antibodies against yourself. Okay? So RH positive is not going to have anti-RH antibodies.
Yeah, that makes sense.
Okay? With RH negative now, do they have anti-RH antibodies?
Not unless it's exposed.
The question, or the thing here, is maybe. The answer here is maybe. Okay. If they have been exposed, they can develop anti-RH antibodies.
Why can that happen here, but
I don't know.
why is exposure necessary?
I don't know. I don't know. And it's because there's more involved in developing antibodies to blood type. There's gut involvement. There's how your immune system is tuned. Okay. Why? I don't know. Okay. You may be able to find it on the internet, and if you find it, tell me. Okay. But okay. For our purposes, that's the way the ABO system is set up, and this is the way the RH system is set up. They're just different. Okay? All right, so an RH negative person, do they have anti-RH antibodies? Only if they've been exposed. So how would they get exposed? Okay, exposure to RH positive blood. How do you get exposed to RH positive blood?
Blood transfusion.
Okay, blood transfusion is a big one.
Trauma if you have a baby who is positive and
And then pregnancy when you have a baby that's positive. Okay, now, you will be asked, okay, on your test and also on NARM, okay, you will be asked 46 ways from Sunday about RH problems. Okay, the bottom line, you will never, ever have an RH problem in a baby unless the mother is negative. Okay? They will tell you the sisters, the brothers, the fathers, the babies, and everybody else's blood type. It doesn't matter unless the mother is RH negative, because it's the mother's antibodies that are the potential problem. Okay? So, when you do your history, do you ask people how many babies they've had? Yes, but do you stop there? No, what else do you ask? How many pregnancies they've had. Okay. Because blood cells are present in the embryo very early. Okay. Now, the baby's not going to have a complete circulatory system for several months, but you do have blood cells that are present in the yolk sac and other places very, very early. So even when a person has had a miscarriage, even when a person has had an abortion, okay, you still need to count that as a pregnancy. Now, that's all well and good, but when we get to gynecology, we're going to talk about how most pregnancies, most conceptions don't take. Okay? So oftentimes the implantation doesn't occur properly, especially in young women who have a fairly irregular cycle sometimes, they may not ever be aware that they were pregnant. They just know that this period was a little heavier. Okay? So, can you have an Rh negative mother come in and say, well, I've never had a baby, and so therefore I haven't been exposed. I've never had a baby and I've never had a blood transfusion, therefore I should have no problems.
No.
Can you say that?
No.
No, because there's no way for her to know for sure. Okay? Unless you go in and test her blood, and there are some people who do that. Okay, if they have religious reasons, for example, to not receive blood products. So what do you do to keep this mother from developing anti-Rh antibodies when she's pregnant? Okay, you give them Rogam. Rogam is a collection of anti-Rh antibodies. Now, does that seem a little counterintuitive?
Mm-hmm.
You're giving them the thing that is going to be a problem. Why are you doing that?
I see. So the body will attack the antibodies.
No. So your body won't make antibodies.
Oh.
Okay? Now, what happens when an Rh negative person encounters Rh positive blood? They go, whoa, foreign. What do we got here to fight off this foreign thing here? And they look around. Wait, I don't have any antibodies. So the body tools up and makes antibodies. Okay, like crazy. Okay. But if your Rh negative person looks out and says, there's Rh positive, what do I have to attack it with? Oh, here's this little package of antibodies here. I'll take that and use it. And they won't make their own. Okay. Now, the amount of anti-Rh antibodies in a Rogam shot is limited. Your body's ability to produce anti-Rh antibodies is practically unlimited. Okay? What Rogam does is it keeps your body from just going berserk and producing all of these bazillions of antibodies. The more antibodies floating around, the more likely they are to cross the placenta and attack the baby's blood if the baby is Rh positive. Okay? Are you going to know whether the baby's Rh positive or not? Without some sophisticated sort of testing, you're not going to know. Okay, now, so they're getting better with figuring out both the mom's blood type, baby's blood type, and being able to do that in ways that are not invasive. Sunny just sent me some stuff on this, which I haven't had a chance to read yet. They're getting better at figuring out how to do that non-invasively. But at this point, you're not going to know whether the baby's A, B, O, Rh negative, Rh positive, whatever. Okay? So you are treating the mom as if it is a worst case scenario. Okay? Everybody understand that?
I have a question
Yes.
about, and this is just, I don't know, I will be way off on how this would work, but if you're giving an Rh antibody, you're giving the antibody to the person's blood, right? Could that trigger an autoimmune? Like, could that?
Oh, wow.
I mean,
autoimmune diseases?
I mean,
The
I don't
triggers,
know.
the triggers from most autoimmune diseases are not understood.
Yeah. I know, well, that's why I'm like,
Maybe that's it. It's possible. The thing, to have an autoimmune disease happen, you have to have a specific genetic configuration yourself, and then you have to have something that breaks your immune system.
I don't understand how the ROGAM doesn't cross over the placenta.
It could,
It's just not enough.
but it's a small,
Small
amount.
amount, so it doesn't.
How much is in a
Yeah.
ROGAM shot? It's what, 2 cc's?
Yeah.
You know, tiny amounts. How much blood do you have circulating? In a
Oh,
normal
yeah.
person, 5 liters, and a pregnant person, more than that.
Yeah.
Okay, so it's a very tiny amount comparative.
Mhm.
Compared.
Okay.
Okay, yes.
at
Wait a
what
minute.
point are the ROGAM shots given?
Okay, now that varies some. Okay, in the past they would give them early and then they would give them late. They would give 2 sets. Okay, why?
They give it before birth to.
Okay, no, they would give it late because when you have the placenta coming off, that's when you have the highest chance of blood mixing occur close to labor,
Usually at 28 weeks.
28 weeks. Why would they give it early?
So that it doesn't.
If you got in a wreck and you think that blood potentially crossed, or if there was
Okay,
trauma.
because things are not perfect with the placenta, okay? And by about 16 weeks, you have the placenta on board and you have baby's blood circulating and, you know, potential for micro tears. Also, if on the early end there, you want to make sure that there's not a developing problem because the baby's got all the rest of that growing to do. Okay, and you know, I say early and late. The recommendations for the number of weeks I've seen change.
So I have a question.
Yes.
So if we're giving such a small amount, is there still a potential that even with ROGAM, a mom could become sensitized? Especially if you have something like before 28 weeks and
Yeah.
then like even after 28 weeks?
Yeah.
So
You
even if
are-
she gets ROGAM, you don't know 100% for
Which
certain
is
her
why-
subsequent.
Which is why they always give ROGAM with each pregnancy.
That's interesting.
Yes.
So as far as I understand, ROGAM is for the D antigen. What about other antigens?
Well, no, ROGAM does not work on anything but the RH antigen.
Okay.
Okay. Now, is your textbook calling that the D antigen?
Yeah.
Okay.
But what if you have a weird type? So like an abnormal type.
Okay. An abnormal type how?
Okay. Well, one of the pregnant women at the birth center has like an odd RH antigen F or something weird. And so she wasn't like, they don't prescribe her ROGAM for that specific antigen. And so it's just like luck, basically. Are you going to?
Okay.
Hmm.
Now, remember, the story that I'm telling here is the basic story. Okay, just to keep everybody's head from exploding. Okay. What she's talking about is that there's sometimes variations in the RH flag. Okay. Would a person develop antibodies against a flag that is different? Yes. Okay. Is ROGAM going to take care of that? No. Okay. It's also why you don't give ROGAM for ABO incompatibility. Okay. And there are other antigens. There's Cal and Delta and however many, there's 15 or 16 other ones just on the blood typing. Okay. When you get into the histocompatibility antigens, there's even more. Okay. So can you have blood mismatches? Yes, but for reasons that we don't completely understand, the other ones don't usually cause as much problems as RH. All right, now, yes, Kitty.
Okay, I'm sorry. One more question. I was just curious because you said this came from rhesus monkeys. Do rhesus monkeys have the same problems that humans do? Like if they're, or do they all carry it?
I
Does that
don't
make
know.
sense?
Or like
Yeah.
miscarriages and stuff.
Yeah.
Retaining pregnancies.
Yeah,
I don't know.
because I'm reading that it's like only in humans do we have this specific problem, But I'm like, that can't be because rhesus monkeys, we discovered this from rhesus monkeys.
I'm not sure whether anybody has cared enough about monkey miscarriages to actually count it.
I know.
But they may have, because rhesus monkeys are used as test subjects a lot. So if you want to dig around on the internet, there's probably somebody out there who's looked. Okay. All right, now, the biggest problem with Rh disease is when the mother's antibodies destroy the baby's blood. Okay, that condition is called erythroblastosis fetalis. Now, when you hear about babies having to have a blood transfusion immediately after they're born, or if you hear about babies having to have a blood transfusion in utero, okay, this is usually what's going on. There's such an antibody response from the mom that the mom's antibodies are killing off the kid's blood cells as fast as the kid can make them. These babies are born and they tend to be blue. Why?
Because their blood's deoxygenated.
Because their blood's deoxygenated. Why?
Because their blood cells are being killed.
Because they don't have enough blood cells to carry the oxygen around. Okay, so they call these blue babies. It's not the only thing that can cause a blue baby. The other things are cardiac malformations. Okay, now let me ask you a question. You have a lady who is Rh negative, and she comes in with her husband, and he is Rh negative.
Very good.
Do you give her a Rogan shot?
No.
Yes,
Yes?
you do. Why?
You still do?
Because it's a not, it could be from former.
Oh yeah, the baby could still
Like,
be
it could
positive.
anywhere in her family.
Or his.
And you
No.
can't guarantee 100% that it's her husband's, even though she said
Well,
and now
there is no way. Is there any way for you to know who fathered that kid?
Oh.
No. Okay. So you give her Rogan. Okay. Now there are some people who come in and they're very well educated and they're up on this and they may have reasons. Okay. The only safe way to do it is to check her antibody status. You can do an Rh antibody titer to see. Okay. And if she does not have anti-Rh antibodies, and if he is Rh negative, and she knows that's the father of her child, and she's willing to, risk that and all that, okay, then in those situations you can allow somebody to do that. Okay? Some of the OBGYNs don't, though. Okay? And you can see why. All right, now, you have an antigen for blood type from your mom, and you have an antigen for blood type from your dad, so you have two. So if you are Rh negative, you have two Rh negative antigens. Okay?
Mhm.
If you are type A, you could have two A antigens, or you could have an A antigen and an O antigen. Right? It would still be type A. If you are type AB, you have an A antigen and a B antigen. Correct? Okay, and so forth. You see how that goes?
Mhm.
So, can you declare positively that a man is a child's father based on blood type?
No.
No. Can you rule somebody out based on blood type?
Isn't there like
Okay,
some kind of
so-
assessment?
Mom has AO blood. Dad has type O blood. Type O, okay. The kid has type AB blood. Okay. Is this guy the father?
No.
No.
Mm.
I thought it was way more complicated than that.
It
though.
is, but-
Okay. Like that.
Okay, so you can rule somebody out, but you can't rule somebody in as a father. she could have, say you had type O dad and type O kid. The father over here could be AO, he could be OO, he could be BO. See that? So there's 3 different guys here. It could be the father of that kid. So you can rule somebody out with blood typing, but you can't rule them in. So you rarely see blood type being discussed with paternity suits anymore. They use DNA, okay, because it's just a whole lot easier to
I see.
line that stuff up. Okay, does that make sense?
Could the dad also be type OB or OA?
Yeah. Okay.
Is O the recessive then on that?
No, they're codominant.
Okay. So then how would they have, if they were AO, how would they just type A blood then?
They're codominant.
Yeah, but are you just specifying
You
just type A?
you name it based on the presence or absence
Okay,
of a
okay.
flag.
Oh, yeah, okay.
All right. All right, let's take a break, and then there's a couple other things I want to finish up on the other end.